Sacchariosis using bioinformatic tools. Next, in silico approaches for designing the beta-lactamase inhibitor and in silico genes and protein network analysis of beta-lactamases in Indian Council of Medical Research. Understanding the mechanism of multi-drug resistance in pathogenic bacteria and identification of potential drug targets. And genomic systems of biology studies to understand the mechanism of multi-drug resistant pathogenic bacterial species of clinical importance. Also, in bioinformatics and molecular approach to categorize the mechanisms of multi-drug resistance in Salmonella typhi recovered from children with typhoid at Pediatric Specialty Teaching Hospital. He has guided 9 PhD students successfully and presently guiding 6 PhD students. He has published more than 130 articles in both national and international peer-reviewed journals. He has delivered more than 40 invited talks, presentation, and academic assignment. It's our pleasure to have you here with us, sir.
Thank you, Sushmita. Now I call Dr. P. Vidya, Head of the Department, PG and Research Department of Microbiology, to felicitate our Chief Guest.
Yes.
Yes.
தமிழுக்கு முதல் வணக்கம். அனைவருக்கும் தமிழ் வணக்கம்.
வணக்கம்.
So it's an honor for me to be here. My Guruji, Vidya Madam, I am here because of her. Whatever I am here today because of my Guruji Madam. In fact, this is what I was telling Madam's colleagues, Dr. Jagdishwari and Dr. Kritika, that if not for Madam, I would not have studied MSc itself. So all the credit goes to Madam, and it's always an honor to be here. And whenever Madam calls me, there's no question of saying no to Madam. So whenever Madam calls, I said, Madam, you tell what needs to be done, we will do it for you. So whatever we are is because of Madam. So thank you, Madam, for this invitation. It's an honor. It's an honor to be here to interact with your students. So, in fact, Madam students is interacting with Madam students directly. So in a way, it's like that. So coming to the topic of today's discussion, opportunities and challenges of microbiology in the present century. We know that microbes existed before us, and we are the last, so-called last in the evolution. If at all we believe in science, we need to believe in it, the theory of evolution. And we are the last race, and we are the most dangerous race among all the species that is known to us. Because we are the only ones who don't bother about environment. We are the only ones who don't bother about other species. We are the only ones who are bothering to damage the other planets as well. Now we are exploring whether to damage Mars or Venus, and to see where, if there is some other planet near Jupiter, there is some water, we'll go there and do all the damage that we have done to planet Earth. And the maximum damage that has been done, if you look at it, it's because of the human race. And whatever damage we do is being rectified by the microbes. So if not for the microbes, the whole planet would have become extinct long back. There would not be any life forms. And as students of microbiology will appreciate the role of microbes in environment, in the preservation of environment and so on. But I belong to a different school of microbiology where we deal with very minuscule number of microbes, which is basically we call medical microbiology, where we discuss about pathogens. But if you look at the number of pathogens that we know is very, very less. That's what I said, it's very minuscule. And we are all in the post-COVID world now. Hopefully, we will be continuing our normal life like this. Instead of having an online class and online presentation, this is much better. Imagine a microbe which cannot replicate on its own, which needs a living cell to survive, and you need an electron microscope to see. You know, if you look at the virus, we have hair. I don't have much hair on my head, but you know the hair. 1 in 10,000 is the size of a virus. தலை முடி இருக்கு இல்லையா, அந்த முடியில 10,000 பாகத்துல ஒரு பாகம். So அவ்வளோ small nano size particle can bring the whole world to a standstill. We say America is very powerful, they can launch satellites, this, that and all. They were helpless. They were helpless. The entire world was helpless. We all know about it. So the nature knows how to balance. For the past 2 years, there is rain everywhere. It's because the environment is good. The cycles, the geological cycles are all happening in a normal way. So nature has got its own way of balancing things, and we need to respect that, and we need to respect the microbes as well. And there are lots of beneficial microorganisms. Even if you look at our own system, the human system, look at the number of cells. I am not terming the size of the cells, the number of cells. If you look at the number of human cells that we have, and if you look at the microbes, number of microbial cells that we harbor, predominantly in the JIT, the number of microbial cells are much higher than our own cells. So if there is an imbalance in the number of cells that we have in our JIT, there will be lots of problems. I don't know whether you would have heard about it. There is also something called a new term that is coming up in a big way, microbiome. And people talk about fecal transplant, which you may not like it, but that is the fact, because that's how you manage or you maintain your normal microbiome levels in your body. So if I'll quickly go through the slides. Okay. So yeah, this is fine. So I did bioinformatics after my master's. So I thought when madam said, madam, I'll also speak on bioinformatics. So what is bioinformatics? No, now we are in the data-driven world. Everywhere, thanks to Mukesh Ambani, we can make free calls. I think you can make free calls. Earlier, when email came, I don't know whether you know or not. Incoming call will be charged for us. Someone will call, be unknown person, missed call, or we invented this call. Because we have to give a call and they will call. So it's all data driven. That's why what I'm telling is all data driven. So managing data has become important, and there is lots of biological data that has been generated. You would have heard about the whole genome sequencing and the various genome projects that happened after the human genome project. And to manage that, it's not possible to use your normal paper, pen and pencil. So take for example your own college, DG Vaishnav. You have got several programs here, both aided, unaided, and there are different programs. Earlier days, they would have maintained lectures and files and so on. But now they have a system. Everything is, there is a database, for faculty, there is a database for students, and there is a faculty student relationship between entity to entity. Likewise, we also have biological databases. We have sequence data, we have protein data, we have metabolomic data, we have pathway data. There are several databases, and we can relate. For example, you take a well-studied bacteria being E. coli. We have a sequence from E. coli. This sequence is related to E. coli. Similarly, who is the scientist who did this sequence? What is the importance of that sequence? Have they published any information pertaining to that sequence? So all those information you can put it in what is called as this database. Since you can relate, we call it as relational database models. Maybe computer science, if you study computer science in detail, you will understand that much better. Okay, can you move on to the next? Yes, this is how it works. This database works. Next slide. Yeah, this is a very good representation. You tie your eyes and you allow a person to study the elephant. Each person will feel one part and he will give one description. So bioinformatics is also like that. For people who are averse to computers or people who are not familiar with computer science, they will think bioinformatics is about all these things. Yes, it's all about all these things. And you can manage all these things by using a simple system. That is what we say. It's very simple in fact, but to understand that it takes time. Next slide, please. So this is what I said. There is lots of information. Now you see, for example, we all have taken a COVID vaccine, either a Covaxin or Covishield. People who are fortunate to have taken a Pfizer or a Moderna vaccine. Now, how is this vaccination being done? And you know the normal process that takes place for a vaccine from the initial trial stage to the final licensing stage, it would have taken around 7 years. Now imagine if COVID, even though the outbreak started in 2019, we had it in the later part of 2020. Imagine, like we say, 2020, we had COVID, this vaccine discovery will take 7 years, 8 years, no problem, let people die. Whoever survives, let them survive. Whoever dies, let them die. Can we leave them like that? Can we leave them like that? Then why should you and me study? Why should we study? We are studying because to serve humanity. And we need to do whatever science that we have studied should be applied. So the vaccine discovery was so robust because of the applications of bioinformatics to a large extent. We have databases, we have robust data mining programs that are available. So with that, we could speed up. Similarly, we know we had antibiotic discoveries. Earlier, it used to take 12 years, but now we can do it within 3 to 5 years. That is also because of the advent of bioinformatics, where we can simulate how an antibiotic works in a system and say this is how it works. Instead of working on 10,000 molecules, we can work on 1,000 molecules. So what it does is it also reduces the cost, it reduces the time, it also reduces the amount of labor, workforce that is required for all the discovery process. So bioinformatics has played a major role in modern biotechnology as well as in modern microbiology. So there are lots of benefits. So as I said, not all microorganisms are harmful. It's only a small number of microorganisms that are harmful, predominantly bacteria, a few viruses. Then we also have a few fungi, which are also along with parasites. Thankfully, there is no algae that is being known to be pathogenic for humans. I hope it remains such that there is no algae that is pathogen. Otherwise, we don't know what the Chinese will do, but hopefully they don't do any research on algae. Otherwise, we might land up in another problem. So, we work on our lab. Two of our students here. We work on antimicrobial resistance. So, what is antimicrobial resistance? Basically, you take an antibiotic to treat a bacterial infection, but the bacteria does not respond to the antibiotic. In the sense, the antibiotic is not able to inhibit or kill the bacteria. That is because the bacteria has developed resistance. So people usually what we talk as people practice science, what we do, we say the bacteria has developed resistance on exposure. Yes, of course it is true, But we should also understand that what is the source of antibiotics? What is the source of antibiotics? Where does antibiotics come from?
Microbes.
That is also basically fungi and actinomycetes. They are also microorganisms. So நம்ம ஊர்ல சொல்லுவாங்க கிராமத்துல எல்லாம் முள்ள போட்டால தான் எடுக்கணும். So to treat a microbial disease, we have to find a solution from a microbial source. Yes, do you agree? So now we discovered antibiotics. Penicillin was discovered by Sir Alexander Fleming, 1923. We all know that. But even before Fleming could discover antibiotics, these two microorganisms existed together. So bacteria, Staphylococcus, E. coli, Vibrio cholerae, Salmonella typhi, Microbacterium tuberculosis, Yersinia pestis, you name any particular pathogenic bacteria, they all existed. Similarly, penicillium existed, actinomycetes existed, Streptomyces existed. So these are the source of antibiotics. So they had an interaction long back. The human race came much later. So there are reports penicillinase was known to us even before penicillin could be discovered. So what is penicillinase? It is an enzyme that cleaves penicillin. So we think that the bacteria started producing penicillinase only after penicillin discovery. It's not like that. Penicillinase was known even before penicillin was discovered. I think Madam taught us microbiology. I mean, when we studied microbiology, Madam was a molecular biology teacher. I still remember the lacopron. The lacopron concept, what is the lacopron concept? When the bacteria is exposed to lactose, the enzyme beta-galactosidase will be produced. There is a switch-on, switch-off process. Only in the presence of lactose, the enzyme will be produced. Otherwise, it has the ability, but it does not need to produce. Similarly, microorganism has got all genes to produce proteins which are capable of cleaving these antibiotics. Only thing is they are not producing it because there is no need. Suppose you are planning to go from TG Vaishnava College to some other place in Chennai. You have a car. You will go by car. You will not go by bus or auto or Uber or Ola, whatever. Suppose if your car has a problem, what will you do? You will look for other means. So similarly, bacteria will also look for other means only when there is no other option. So when there is an option, why to produce unnecessarily? Suppose you have a car, your car is functioning fine. Will you leave your vehicle in college and go by auto or bus? We are fooling ourselves. So bacteria will not do that. They are much smarter. Please understand, we think we are smart. It's not true. Bacteria, microorganisms are the most smartest living species known. They know how to survive. They know how to balance. We have to survive in their presence. We have to survive among microorganisms. We have to learn how to survive, how to deal with them. It's our learning. So what we do is in bioinformatics research, we might say that our molecules or our compounds will lead to some new discovery. All these are fine. This is basically to convince the scientific community. What we should understand is how is it the bacteria is developing resistance? What is the probable mechanism of resistance? So that is what we try to find out. And our course of research for the past 1.5 decades is basically understanding the mechanism of antimicrobial resistance. And our scholars have done a lot of work on that aspect. And I'll slowly go through what we have done so far. This is very important before I go to the mechanism of antibiotic resistance and understanding how it happens. This is very important. See, I hope all of you are microbiology students. Please understand that antibiotics should be taken only when you have a bacterial infection. You have a normal cold, you go to a doctor, fine, no problem, he prescribes antibiotics, please don't take. You take all these antihistamines, anti-inflammatories, all that is fine. But please don't take an antibiotic unless until you have a bacterial infection. Indiscriminate use of antibiotics is a major reason for antibiotic resistance. So take antibiotics only when it is prescribed for bacterial infection. Complete the entire course. For example, your doctor says take it for 5 days. You will take for 1 or 2 days, then your problem will subside. You will say, okay, now the problem is subsided, so I will not take. So when you do that, what happens is you are enhancing the bacteria to develop resistance or to exhibit resistance. And my doctor prescribed medicine. 2 days it got cured. I will preserve the medicine, put it in the box. If I have the same symptoms next time, I will use. That also should be avoided. Please educate your parents, your friends, everyone. Do not take antibiotics for viral infection because it will not work for viruses. Do not use your friends' antibiotics, your father's antibiotic, mother's antibiotic, grandma's antibiotic. That is not be done. This I think all of you will be knowing various antibiotic targets, right from cell wall, cell membrane, the nucleic acid, as well as the protein biosynthetic pathway. So this is how we have several classes of antibiotics, and each class will have its own action. Go to the next page, please. This is the problem. This is the problem which is existing even before antibiotics could be discovered. So what are the problems? One is basically, an antibiotic, take for example penicillin. If it has to enter into the bacterial cell, it has to go and bind to something. So what is the binding receptor? It's basically the penicillin binding protein. There are some antibiotics which go through the cell membrane, the bacterial cell membrane. So for that, we have what is called as the membrane channels, either through the porins or through membranes. And there are other antibiotics which go and bind to either the nucleic acid or to the ribosomes, basically for the protein synthesis. So how does this bacteria develop resistance? One is, with respect to penicillins and cephalosporins, the major mechanism is through binding with penicillin binding proteins. So instead of binding it, it will repel. So how does it repel? It changes the receptor. Take for example, I think you all study immunology as well. The antigen antibody reactions are highly specific. So likewise, this antibiotic binding with the receptor is also specific. Suppose if you want to catch a apple, you have to have your hand right to hold the apple. Similarly, oranges, whatever you want. So you will have to have some specific receptors, specific shape and position. So when it alters, so suppose if I think all Indians are crazy about cricket. So instead of the batsman hits ball in the air, the fielder has to catch it. So the holding position and all is important. So if the fielder is not holding the hand in the proper way, drop. So likewise, the bacteria also alters its receptor so that instead of the antibiotic going and sitting in the target, it will slip it. So once it is binding only, the bacteria will be inhibited. Otherwise, the bacteria will not be inhibited. So it is altered. Similarly, the cell membrane, it has to penetrate into the cell membrane. It alters the permeability of the membrane so that the antibiotic cannot penetrate. So we try to do something and then put it inside. What it does is it afflicts box. We have what is called as afflicts box. It's just throwing the enzyme, throwing the antibiotic outside the cell. Even that we overcame something, and then what happened is we came up with what is called as the inhibitor combinations. So penicillin, cephalosporins with clavulanic acid, sulbactam, azobactam, all these inhibitor combinations came. Then cell membrane also we managed, we put it inside, afflictions also we stopped. In spite of all these things, to make the antibiotic present inside the bacterial cell, the bacteria said, okay, no problem, let it be inside, I will produce an enzyme that will cleave it. So that's how we had what is called as beta-lactamases. But now we are living in an era of extended spectrum beta-lactamases. So what is the difference between beta-lactamase and extended spectrum beta-lactamase? Beta-lactamases will act on only the penicillins and cephalosporins, whereas ESBLs will act on both antibiotic penicillin, cephalosporins as well as on the inhibitors. That is what we call ESBLs. Yes, these are all publications. Before going into publications, the faculties understand the importance of publications. But as students, I need to tell you what is the publication. இப்போ நம்மெல்லாம் ஒரு நியூஸ் தெரியுதுன்னா என்ன பண்ணுவோம்? ஏதாவது ஒரு செய்தி நமக்கு தெரியணும்னா என்ன பண்ணுவோம்? We watch news. தொலைக்காட்சி பார்ப்போம், இல்லைன்னா நியூஸ் பேப்பர் படிப்போம். ஒரு சயின்டிஃபிக் டிஸ்கவரி தெரிஞ்சுக்கணும்னா என்ன பண்ணுவோம்? How do you know that there is a scientific discovery or invention? To articles. So what are these articles? basically researchers they do something and they publish. So it's not like that you do some research and you send it to some journal, they will accept. They are all peer-reviewed international journals, which means there are peers. Peers means நம்மள மாதிரி researchers, scientists who are accomplished in that field. They will check our paper and see whether there is some merit in accepting this manuscript that we have submitted for publication. So all these papers that we do, maybe research scholars, if you are here, you will understand the importance. So all these, whatever I am presenting here, is basically the peer reviewed, been reviewed, revised, and accepted. So what we did earlier was basically to understand how these beta-lactamases and penicillin binding proteins bind and how do they cleave. This is what we did, and we could produce these papers. First author is proud student of Madam. I am standing here. Our students are also doing well. K.M. Kumar is a scholar who was a scholar with us some time back. Now he is an associate professor in Central University of Pondicherry. Next one, please. Okay. How many of you like biochemistry? Please raise your hands. Do you like biochemistry? Most while I was a microbiology student, I never used to like biochemistry. I used to ask why I should study biochemistry. It's not like that now. My wife is a biochemist. That's a different story. But biochemistry is very, very important. Identification of microorganism. How will you differentiate Staphylococcus aureus from other Staphylococcus? It's a Coagulase test you do. You do a Coagulase test. You say if it's Coagulase positive, you say Staphylococcus aureus. If it's Coagulase negative, you say it is a Coagulase negative Staphylococcus, which means other than Staphylococcus aureus. So, identification of microorganisms also is dependent on the biochemical test. So, biochemistry is very, very important. So, in the context here, it's basically the biochemistry here. It's the structure of proteins. You would have studied the structural organization of proteins. When the protein is in the linear N-terminal to C-terminal, we call it is a primary structure. The primary structure can either fold. It folds in the form of a helix, coil, Mariana, we call it as helices, spring Mariana, helices. Pleated sheets, we say it is the secondary structure, beta pleated sheet, beta strands and so on. Helix and strand intermediate, we say it as turns, coils or bends. So there is folding. And this helices, coils and bends, whatever we call the non-regular secondary structure element, it superfolds and forms what is called as a tertiary structure. So very quickly, I am not going into the detail of biochemistry. Very quickly, I will tell you something which is very, very important. A sequence, N-terminal one position. C-terminal last position. Or amino acid, or protein, it's all made of acids. You know that there are 20 different naturally occurring amino acids. So first position to maybe take over 150 amino acid residues. There is a protein with 150 amino acid residues from 1 to 150. So the sequence question, it's in the order. Neighboring, 1, 2, 3, it's all like. In the protein folds, what happens is in the secondary structure, they are all neighboring. And when it super folds, what happens is a fifth residue, amino acid in the fifth position, will come closer to the 100th residue. The sixth residue and the fifth residue may be very far. This is what I am saying in the three-dimensional state. So the protein, if it has to be functionally active, it has to be in a 3D state. That is why the three-dimensional structure is important. So when understanding the three-dimensional structure, there are several molecular interactions that we study. For example, the hydrophobic interactions, electrostatic interactions, disulfide, covalent bonds, all these. In addition to these, now we have what is called as a non-canonical hydrogen bonds or non-covalent hydrogen bonds, and so on. And those interactions are also important. Now, what we did was we studied these interactions in two classes of important proteins. One is the penicillin binding protein, the other one is the beta lactamases. So penicillin binding proteins are important for binding antibiotics. Beta lactamases are important for cleaving the antibiotics. So what is the difference in the non-covalent interaction patterns? So we did that, and the subsequent slides. And these are some of our papers of that. See, Lavanya was a PhD scholar. So she published papers. She was in Detroit for some time. Yes, these are all Lavanya's papers. You can see. Yes. This is a very interesting study. Adan was our student. He's a scientist in Jipmer, Pondicherry now. Very, very serious infections. You know, you would have heard about a patient in a very serious condition. He goes to an hospital, and the doctor will say the patient is a sepsis condition. Or after sepsis, there is coma. After coma, there is death. So, what is the sepsis? Normally, our bloodstream, it should be sterile. There should not be any bacteria inside the blood. Presence of bacteria is what we call as bacterial. So during the bacterial stage itself, we'll have high fever, we take antibiotics, as well as our normal immune system will try to eradicate the microbes and bacteria from the bloodstream. But when the bacteria overwhelms our immune system, and when the bacteria overwhelms the antibiotics, we end up in what is called as septicemia. So what is the difference between bacteremia and septicemia? Bacteremia is a mere presence of bacteria in the blood, whereas septicemia is a condition where the bacteria is present as well as it proliferates, multiplies actively in the bloodstream. And if it happens to be a gram-negative bacteria, it is even more worse. You know, bacteria, the gram-negative bacterial cell wall, if you can go back to your basic microbiology, bacteriology, it has got what is called as a lipopolysaccharide. There is a lot of lipopolysaccharide. They are basically endotoxins. So what are endotoxins? Toxins which are normally present in the bacterial cell. On lysis of the bacterial cell, they are released. Exotoxins are produced when the bacteria is alive. These endotoxins are released when the bacteria lysis. So due to the accumulation of these bacterial cells, there is also death of the bacteria, which results in endotoxins as a result of it. Endotoxic shock. So most people who get admitted for sepsis often result in coma and death. So this study, why I'm saying all these things is to tell you, this study was done in all sepsis patients. People who had very, very severe infection, where the survival rate was very less. First, COVID or the mortality rate was only 0.2%. 100 பேர் கோவிட் வந்தால் ஒருத்தர் தான் சாவாங்க. Mortality rate of bacterial sepsis is more than 90%. Which means what? 100 பேர் செப்சிஸ் வந்துச்சுன்னா 90 பேர் பழக மாட்டாங்க. கடவுள் தான் காப்பாற்றுன்னு சொல்றாரு டாக்டர். கடவுள் காப்பாற்ற மாட்டார், கடவுள் கூப்பிட்டுப்பாரு. மற்றவங்க சொந்தக்காரங்க தான் கஷ்டப்படணும். So it's a very, very serious infection. So we had around 900 samples. It was a pan-India study. Anand Manogaran was here. I think he was here. He was my senior. He was in DG Vaishnav some time back. Same children. We did a study. They did the experimental part and we did the bioinformatics part. They wanted to understand the mechanism of resistance through this OXA. There are several genes, don't worry about it. Oxacillin now much long. It's an penicillin derivative. So OXA was initially discovered, OXA group of genes was initially discovered with oxacillin resistance. That is why it is called as OXA. And we did the bioinformatics study, fully blinded study. Our bioinformatics findings helped them to understand the mechanism of resistance. And we were able to publish this paper. This is PLOS, and again, Mycobacterium. What is the vaccine that is given for Mycobacterium tuberculosis? BCG, sir. Very good. BCG, Bacillus calmette-guerin. Do you know what is the efficacy of this vaccine? இப்போ டிபி நம்ம எல்லாரும் டிபி வேக்சின் எடுத்துருக்கோம், பிசிஜி எடுத்துருக்கோம். அண்ட் பிசிஜி இஸ் கிவன் அட் பர்த். அப்படின்னா நம்மலாம் பிறக்கும் போதே நம்ம ஊசி போட்டோம். நாமெல்லாம் குழந்தைய இருக்கும்போது கத்திருப்போம். எல்லாரும் இப்போ இருக்கிற குழந்தைகளும் கத்திட்டு தான் இருக்காங்க. பட் டு யூ நோ வாட் இஸ் த எஃபிகசி ஆஃப் திஸ் வேக்சின்? எனி கெஸ்? எஃபிகசின்னு வேக்சின் எடுக்கிறாங்க, இவ்வளோ பேருக்கு ப்ரொடெக்ஷன் இருக்கும். கோவிட் வேக்சின் எடுக்கணும்னா கோவிடோட எஃபிகசி, கோவிட் வேக்சினோட எஃபிகசி இஸ் அரௌண்ட் 90%, 95%. What is the efficacy of this vaccine, BCG? Good. Okay. At least I appreciate you for answering. Good. There are two studies done by our own ICMR, Indian Council for Medical Research. One in Kasauli and one in Chengalpet. Chengalpet Medical College. Chengalpet Medical College was one part of the study, and one was done in Kasauli, north-south study. Kasauli study found that this BCG vaccine was found to be 40% efficacious. 40% என்ன 100 பேர் வேக்சின் எடுத்தாங்கன்னா 40 பேர் ப்ரொடெக்ஷன், மீதி 60 பேர் ப்ரொடெக்ஷன் கிடையாது. தட் இஸ் மச் பெட்டர். செங்கல்பட் ஸ்டடியோட ரிசல்ட் தெரியுமா உங்களுக்கு? நம்ம சொல்ல வெள்ளக்காரன் போய் பேச மாட்டான், ஆனால் வெள்ளக்காரன் இப்போ அவன் பண்ண ரிசல்ட் தெரியாது. நம்ம ஊர் ரிசல்ட் தான் கரெக்டான ரிசல்ட். யூனோ வாட் இஸ் த ரிசல்ட் ஃப்ரம் செங்கல்பட்? 0%. ஒருத்தர் கூட ப்ரொடெக்ஷன் கிடையாது. இட் இஸ் ஆல் டாக்குமெண்டட். நான் கதையெல்லாம் சொல்லல. போய் நீங்க பாருங்க, இப்போ பணிக்கிறதுல இருக்கும் மைக்ரோபாலஜி, மெடிக்கல் மைக்ரோபாலஜி பணிக்கிற எடுத்தீங்கன்னா அதுலேயே இப்போ ரீசன்ட் எடிஷன்லாம் போட்டிருக்கான். நாங்கள் படிக்கிற காலத்தில் ஜேர்னலில் இருந்துச்சு. தட் இஸ் ட்ரூ. But you might ask me another question. Sir, we know it's a 0% efficacy. Why is the government still going ahead with this vaccination? It's another question. But one good thing about this BCG vaccine is, even though it is not protective, this is the only vaccine which has got a very, very, very minor side effects. இந்த வேக்சின் எடுக்கிறதுனால உங்களுக்கு பிரச்சனை வருதுன்னா கிடையவே கிடையாது. அந்த மாதிரி மேபி ஜுரம் வரும், அதெல்லாம் நார்மல். there will be an immunological reaction. Vaccine பிரச்சனை, there is no side effects for this vaccine. So that is why government is also taking a chance, even though it is not efficacious. If at all it protects, let it protect. So that is the logic on which this vaccine is given. There is no vaccine, and இது டிபிக்கு ட்ரீட்மெண்ட் தெரியுமா உங்களுக்கு? ஒரு யாருக்காவது டிபி வந்துச்சு என்ன பண்ணுவாங்க தெரியுங்களா? இப்போ டைஃபாய்டு வந்துச்சுன்னா ஒரு மாத்திரை எடுக்கிறோம், காலரா வந்துச்சுன்னா நிறைய தண்ணி குடிப்போம், முடியலன்னா ஓஆர்எஸ் போடுவாங்க, சரியா போயிடும். சிகலோசிஸ் வந்துச்சுனாலும் ஆன்டிபயோட்டிக் எடுப்போம், ஓஆர்எஸ் போடுவோம். வேற எந்த பாக்டீரியல் டிசீஸ் வந்தாலும் ஆன்டிபயோட்டிக் போடும். How is this Mycobacterium tuberculosis treatment different from other bacterial? மற்ற பாக்டீரியாக்கெல்லாம் ஒரு மாத்திரை போதும், இந்த பாக்டீரியாவுக்கு ஒரு மாத்திரை வேலை செய்யாது. You have to take 5 different antibiotics. மற்ற வியாதிக்கெல்லாம் 3 நாள் 4 நாள், 3 days 5 days மேக்ஸிமம். இதுக்கு மினிமம் 3 மாசம், மினிமம் 3 months, மேக்ஸிமம் இஸ் 12 months வரைக்கும் you have to take 5 different. ஒரு நாள் ஆன்டிபயோட்டிக் சாப்பிட்டாவே மறுநாள் நமக்கு தான் வயிறு கலக்கும். 3 நாள் எல்லாம் நம்ம ஆன்டிபயோட்டிக் சாப்பிட மாட்டோம். will not take antibiotics. So I don't know whether you would have seen, முன்னாடி எல்லாம் பார்த்தீங்கன்னா பஸ்ல எல்லாம் பார்த்தீங்கன்னா டாட்ஸ்ன்னு போட்டுப்பாங்க, DOTS. டிபிக்கு வந்து the treatment option that is recommended by government of India is what is called as DOTS. DOTS வந்து directly observed treatment strategy. ஒரு வீட்டில் டிபி ஆக இருந்துச்சுன்னா அந்த ஏரியாவில் இருக்கிற ஹெல்த் ஒர்க்கர் போயிட்டு மாத்திரை கொடுப்பாங்க. மாத்திரை கொடுத்து அவங்க முழுங்குற வரைக்கும் அங்கேயே நிற்பாங்க. இவர் என்ன பண்ணுவாரு, சைட்ல வச்சுப்பாரு, அங்கே வச்சுப்பாரு, இங்கே வச்சுப்பாரு, மாத்திரை முழுங்க மாட்டார். ஆனா அவங்க முழுங்குற வரைக்கும் கையில மாத்திரை உள்ள போற வரைக்கும் பார்த்துட்டு தான் போகணும். அதுதான் டாட்ஸ் ஸ்கீம். அதெல்லாம் பண்ணி தான் இது ஓரளவுக்கு டிபியோட லெவல்ஸ் குறைஞ்சிருக்கு. பட் ஆஃப்டர் கோவிட் திஸ் டாட்ஸ் ஏன்னா வெளியிலே போகக்கூடாதுங்கிறாங்க. எப்படி டாட்ஸ் மட்டும் எப்படி போவார், ஹெல்த் ஒர்க்கர் எப்படி போவார்? கோவிட் இருக்கான்னு தான் பார்ப்பார். டெம்பரேச்சர் இருக்கா, உங்களுக்கு இருமல் வந்துச்சா, ஜுரம் வந்துச்சான்னு தான் பார்த்துருப்பாங்க. உங்க வீட்டுக்கெல்லாம் வந்திருப்பாங்க ஹெல்த் ஒர்க்கர் கூட கம், தேவோட செக் டெம்பரேச்சர், தேவோட ஆஸ்பத்திரி, காஃப், ஃபீவர் இதெல்லாம் கேட்டிருப்பாங்க. ஸோ தேவர் diverted for other purposes. Now the TB has raised after COVID. So the context why I'm saying is, இது பார்த்தீங்கன்னா டிபியை பற்றியே பேசிட்டு இருக்காருங்க, வேற ஏதோ பாக்டீரியா போட்டு வச்சிருக்காரு. இது Mycobacterium phlegmatis. So how is this related to Mycobacterium TB? Mycobacterium TB, how is it different? நீங்க எல்லாம் மைக்ரோபாலஜி ஸ்டூடெண்ட்ஸ், so you should answer this question. What is the generation time for E. coli? 20 minutes. Fantastic. Very good. Super. Madam students improve on that. What is the generation time for Mycobacterium tuberculosis?
72 hours.
Yes, excellent. Just 24 hours. ஒரு பாக்டீரியா இன்னொரு பாக்டீரியா மல்டிப்ளை பண்றதுக்கு 24 மணி நேரம் ஆகும். ஸோ இப்போ நீங்க இதுல எம்டிபி பத்தி ரிசர்ச் பண்ணணும். If you want to do some research, it will take many, many years. So can we wait for many, many years? We have waited for so long. This TB is known to us from Egyptian mummies தான் எடுத்திருக்காங்க MTB-யோட DNA. So can we wait for so long? We can wait, but many people will die. So we should have some model. So Mycobacterium phlegmatis is a model organism for Mycobacterium TB. Whatever you do in Mycobacterium phlegmatis can also be replicated or mimicked in MTB. So this is a rapid grower. இது வந்து ஈக்குவலை மாதிரி சீக்கிரம் வளரும். And you can grow this Mycobacterium speckmatis in a normal media, simple media. Low Jensen மாதிரி specialized media therapy. So we did that, and this was a model, and we could publish several papers. So these are all other papers, as you can see here. 3 months தெரியல எனக்கு. So these are all high-end work that we did in our lab. This person. Okay. Whatever we have seen so far is with the protein. You know the genetic code. There is something called DNA. Transcription happens. mRNA translates into a protein. So whatever we have seen so far is protein. So we thought, why not the gene? Why not the genome level? So that is where the gene network. That makes simple. The gene network ரொம்ப simple தெரியும். They are not very simple. They are very, very complex interactions. To study those complex interactions, we have developed what is called as a gene interaction, so that it's easy for us to understand and learn what is the real reasons for mechanism of resistance. So I would say, proudly but humbly, we are the first group in the entire world to publish papers on antimicrobial resistance using gene networks. Gene networks used only, we are the ones to produce papers. We use the tools developed by someone else, and one of our students who worked with gene networks now is going to NIH, to a place where they develop tools for understanding this. In fact, those scientists were very, very surprised to know that they did not discover the tools to study AMR. They discovered the tools to study cancer. We use those tools to study AMR, and they were very surprised, and now he is going as a postdoc. One of our students, Shravan, is in IIT Mumbai now. Very soon he will be joining NIH, to the people, to the group where they develop the tools for gene network. So these are some of our papers, Anita's papers. Initially we did the initial papers. Then Aniket, Shravan will carry on. Ritika here, sir work. So these are all some of the டெங்கு. நம்ம ஊர்ல டெங்கு வந்தால் என்ன பண்ணுவாங்க, நிலவேம்பு கஷாயம் கொடுப்பாங்க. பட் ஏன்னா one thing which you should understand is, whatever antibiotics, all these things are done, it's all good, it all works, there's no doubt. பட் இதுக்கெல்லாம் முன்னாடி எல்லாம் நம்ம மனுஷங்க வாழ்ந்துட்டோம். Civilization, if you look at civilization, we might have 1,000 reasons, differences with Chinese. One thing is common is we are the most ancient civilization. Indian civilization and Chinese civilization. We had several formulations, other things. Sushruta, I don't know whether you have heard of Sushruta, the father of surgery. And likewise, there were several things. Only thing where we did not do was, we did not document things. Document, and moreover, it was kept as a secret. One thing that is different between us and the Europeans is everything they document. Whereas Indians, if we know something, we keep it as a secret and we don't pass the information to others, so that there is no improvement in the system. So அதனால வர்றது. பட் இதெல்லாம் வந்து இப்போ வைரல் டிசீஸ் எடுத்தீங்கன்னா, கோவிட்கே கூட எடுத்தீங்கன்னா, there is no antiviral agent. Similarly, any other virus, very few. For example, HSV, herpes simplex virus, acyclovir, it will work definitely. Cytomegalovirus, ganciclovir, it will work. But other viruses, there is no new HIV, the antiretrovirals that we have, they will control the replication, but there will be no cure. Whereas with viruses, there are some herbal formulations that will work. So what we did was, we did some bioinformatics analysis, and we found this is how these formulations work. Okay, these are all something. Okay, let's pass it on. Pass it. Yes, yes. சரி, பாருங்க, எல்லாம் அமெரிக்காவுக்கு உங்களுக்கு என்ன பண்றதுன்னு தெரியல இந்த கேஸ். இந்தியா இருக்கும், எங்கேயாவது பாருங்க, நம்ம ஆளுங்க எங்க இருக்கும் பாருங்க. பிஐடி இருக்கா? அமெரிக்காவில் இருக்கிறவங்களுக்கே வழி தெரியல, நாங்கள் தான் சொல்லி கொடுத்தோம் வழி. எல்லாம் இந்தியானா யூனிவர்சிட்டி பெரிய பெரிய நம்ம ஆளுங்க உடம்பு சரியில்லை, நேரம் அமெரிக்கா போயிடுறாங்க. சோனியா காந்தி ராகுல் காந்தி எல்லாம் அமெரிக்காவுக்கு தான் பேசிட்டு போயிடுறாங்க. அவங்களுக்கே ஒன்று. So we did some bioinformatics analysis, and we helped them, and procedures they have long got target. Okay, COVID also we did some work. They're all post-COVID work. Whatever I have presented is because of these people and their seniors. So all the credit should go to them. I hope one day all of you will also make Madam proud as I'm planning today. You'll also end up in a lab where you will be doing high throughput research and you'll be able to produce papers and the world should recognize. So I wish you all the very best.
Thank you, sir.
Congratulations on joining DG Vaishnav. All the best. Thank you.
Any questions?
Okay. Yeah. Permission.
Sir, if one wants to pursue bioinformatics as his career, what are the skills needed?
You know to switch on a computer?
Yes, sir.
More than sufficient. Copy paste பண்ணுவீங்களா? More than sufficient. You know what is a DNA sequence? More than sufficient. You know what is a protein sequence?
Yes, sir.
இது இருந்தா போதும். You will become a successful biologist.
Selected.
Once you know what is the DNA, what is the protein sequence, you just have to interpret using your biological knowledge.
Okay.
Thank you.
Okay.